All right, so our next speaker today is Ailish Ullmann. Ailish took a winding path to Scripps with a background ranging from science communication and social media marketing to scientific diving for kelp restoration. She's deeply passionate about science-based solutions to climate change and biodiversity loss and enjoys sharing this passion with anyone who will listen, and quite a few people who do not want to listen. She has enjoyed spending a year immersed in marine scientific research and, as often as possible, the Pacific Ocean too. The name of Ailish's presentation is Rock Bottom, the effect of hard substrate type and environment on macrofaunal colonization in the Southern California borderland.
Thank you so much for that introduction, Samantha. Bless you. The deep sea is often the villain in our stories. It's the scary, fanged creature in the dark, the terrifying monster of myth and legend, or maybe just the gross, spooky fish that we kind of don't want anywhere near us. And yet, despite this cultural aversion to the deep sea, the deep ocean provides us with so much. Our food is increasingly coming from the deep sea, if not directly, like this delicious Chilean sea bass, then indirectly, as migrating deep sea fish provide food for other trophic levels. The deep sea also acts as a carbon sink, bearing carbon in sediments and rocks and preventing our rapidly warming climate from warming even more rapidly. Medical breakthroughs also frequently come from the deep sea, as bioprospectors have found that some deep sea animals are really good at preventing bacteria from settling on them, making them perfect to mimic in creating antibiotics and anti-fouling agents. Increasingly, it looks like we may get our green energy transition from the deep sea as well. The deep sea is home to mineral-rich hard substrates whose contents may be mined for the electric vehicles, solar panels, and wind turbines of a carbon-free future. But of course, all of these ecosystem services come with a cost too. And the result is that we are adversely impacting the deep sea faster than we can effectively study it. Hard substrates, meaning basically anything animals can live on that's not soft and squishy like sediment, are of particular interest, especially since so many of them are being targeted for exploitation by deep sea mining or being harmed by fishing practices like trawling. However, there are significant knowledge gaps in deep sea benthic ecology, particularly for smaller animals and non-reducing ecosystems, meaning areas without vents, seeps, or carcass falls. Most hard substrate research is on these reducing systems, but the hard substrates in between may serve as stepping stones between these hotspots of deep sea abundance, making them very important. Ultimately, we need to understand the connectivity and resilience of these systems in order to properly manage them. How and to what extent are these systems connected? How resilient are they to disturbance and change? How important is substrate type in determining what communities live where? Which brings me to my project. My project took place here in the Southern California borderland. This is a region of high habitat heterogeneity, meaning there's lots of different hard substrates for animals to live on, including basalts, carbonate rock, mineral-rich rocks like phosphorites and ferromanganese rocks, and due to its position near shore and along whale migration routes, carcass falls and wood falls are also common in this region. As a result, it is a living laboratory perfect for studying the impact of different substrates on deep sea animal communities. Lisa Levin's lab conducted an experiment here from 2020 to 2021 that placed different hard substrates out at 2 different sites to study the effect of proximity to shore and 2 different depths to study the effect of oxygen. The inshore site was 40 Mile Bank. Being inshore, it was expected to have higher particulate organic carbon, or POC flux, to the seafloor, as primary productivity is higher closer to shore, and more primary productivity on the surface means more food sinking to the depths for deep sea critters to eat. The offshore site was San Juan Seamount and was expected to have lower POC flux to the seafloor. Each site had a shallow deployment, around 700 meters, which was right in the middle of the oxygen minimum zone, which is a band of deoxygenated water that is present in all ocean basins. The other deployment was deeper, at the edge of the oxygen minimum zone, so slightly higher oxygen, at just over 1,000 meters deep. The substrates deployed at each site for this 10-month colonization experiment included wood blocks, carbonate rocks, mineral-rich rocks like ferromanganese or phosphorite rocks, and sedimentary rock. The substrates were left at these sites for 10 months to see what would move in or colonize these different substrates. That basket in the middle is full of bones, which were part of a colonization study, just not this one. And background rocks like these ones were also collected alongside these experimental substrates to allow us to compare the number and kinds of animals that moved into the experiment to the types of animals living on these types of substrates naturally on site. Once collected, the substrates were rinsed, animals removed, and the substrates were measured and photographed. The next step is where I came in. I sorted each sample of collected animals for macrofauna, or animals retained on a 300 micron screen. For those of you not terribly familiar with microns, this is essentially animals that are visible to the naked eye, but that need a microscope to be able to see more clearly and identify. So I sorted all of this macrofauna into major taxonomic groups and then counted them all up. It took a really long time. The purpose of conducting this colonization experiment was to observe how macrofaunal assemblages differ or share similarities between hard substrate types, whether assemblages differ under varied environmental conditions from the two different sites and depths, and the capacity of macrofauna to colonize varied defaunated hard substrates across these differing environmental conditions. Some of my main questions, though certainly not all of them, were, do density and community composition differ across substrate type and environmental conditions like oxygen, depth, and proximity to shore? And how do composition and density on experimental hard substrates compare to those found naturally on background rocks? So now that we are nicely oriented, let's dig into some results. After sorting over 25,000 animals, I found that macrofaunal assemblages were distinct across substrates. So this was especially true for wood substrates, which were dominated by a mollusk called Xylophaga, but it was also true across the non-organic substrate, with each type of rock exhibiting different high-level community composition. For the wood substrates, Xylophaga was so dominant that it masks some of the other trends exhibited by other animals in these samples. When Xylophaga are removed from this plot, we see that almost all of the mollusks in those samples were Xylophaga, and that there was actually an incredibly high percentage of annelids making up the remaining animals. And this is because those Xylophaga are a pretty special animal. Xylophaga are considered early successional stage animals and habitat engineers because they are generally the first animals to move onto wood substrates. This is because they have a symbiotic gut bacteria that allows them to digest lignin where other animals can't. And that's why their name literally means wood eater. This is what they look like. When Xylophaga move onto a wood substrate, they bore into the wood and create habitat for other animals. Also, as they munch away at the wood, they create wood fragments and fecal matter that other opportunistic animals can use as a source of food. And this is why it's common to see opportunistic taxa like annelids, which are worms, move in right behind these bivalves. And because of Xylophaga's ability to rapidly and abundantly colonize wood substrates, they were the main drivers behind a lot of my results, including density trends. Densities were significantly higher on wood substrates than on other substrates, exclusively because of Xylophaga. With these bivalves removed, densities did not differ significantly between different substrates. There's actually so many Xylophaga in these wood samples, the scale of the density differences between the other substrates is pretty difficult to see. But when we remove the Xylophaga densities, it's a bit easier to see that while the densities of these other substrates do differ somewhat, they do not differ in a statistically significant way, and indicate that while animal assemblages may have been different across substrate types, the density of these animals were not. As far as variation across environmental conditions go, with Xylophaga included, there was no significant difference between sites, depths, or oxygen levels. And this is because those Xylophaga seem to have masked the other trends in the data. Xylophaga tend to prefer shallower depths, and generally, if there is wood around and they are able to, they will come and colonize it. Whereas the other animals exhibited some different trends. When we took the Xylophaga out of these density calculations, we found that there were significantly higher densities closer to shore at 40 Mile Bank, and this is likely because of the higher POC flux to the seafloor there, so more food availability for these animals. There was also significantly higher densities at deeper deployments, likely because there's more oxygen availability there. Different taxonomic groups also exhibited different trends. Annelid densities were larger closer to shore and at higher oxygen levels at greater depths, so following those trends I just spoke about. Echinoderm densities were also larger closer to shore and on non-organic substrates. Most of the echinoderms that I found in this study were ophiuroids or brittle stars, and it is well known in the literature that these brittle stars tend to rely pretty heavily on POC flux or on marine snow for their food source, so seeing more of them closer to shore sort of aligned with that research. And Xylophaga densities were, of course, highest on wood, and in fact, Xylophaga were exclusively found on wood when we're looking at the healthy adult Xylophaga. There were a much smaller amount of Xylophaga found at the offshore shallow site. This sort of reduction in density was not statistically significant, but I did find it notable. And it's probably due to environmental conditions that were not studied in this particular experiment. Things like high water turbulence or high flow rates that's often seen at seamounts. More research would be needed to confirm that hypothesis, however. Finally, densities were higher on colonization experimental substrates here on the left than on the natural in situ background rocks that were collected for comparison. Once again, we can see that those wood substrates have way higher densities, not only than the background rocks, but also, of course, our colonization rocks. So taking the wood out to better compare the non-organic substrates, we can see that those densities are significantly higher on the colonization substrates than on the background rocks. So moving on to some conclusions. First and foremost, different substrate types hosted distinct assemblages. And in fact, substrate type appeared even more impactful than environmental factors in determining these assemblages. Xylophaga, of course, facilitated wood substrate colonization by other taxa, particularly annelids. And even apart from wood, there were distinct assemblages on each of the different rock types. And in fact, some of these assemblages resembled those naturally found on these substrates out in the wild. So woodfalls were densely colonized by Xylophaga and opportunistic annelids, and carbonate rocks, which are formed at and by methane seeps naturally, saw high densities of annelids and crustaceans, not unlike carbonate rocks that would be found at an inactive methane seep. These substrates in their natural form are often associated with highly productive biodiverse hotspots in the deep sea. So how quickly these animals in the experiment were able to find, settle on, and colonize these substrates could have long-term implications for our understanding of the resilience of these hard substrate systems, as well as the connectivity between them. Finally, the higher densities on colonization substrates may indicate earlier successional stages than exhibited on background rocks. In other colonization experiments elsewhere, albeit at reducing systems, colonization to the point of reaching background levels tended to take years to decades. And so it may just be that these were fresh habitats that these animals rushed into, and given enough time, they would start to resemble the background assemblages and densities. However, it is also possible that short-term environmental changes may be responsible for the higher densities on colonization substrates. The background rocks that I used for comparison were collected at the beginning of the experiment, and we have not yet analyzed the background rocks that were collected at the end of the experiment. So analysis of those final background rocks will be necessary to determine if there are any changes in seasonality, for instance, that may have impacted these results. Ultimately, more deep-sea research is needed across ocean basins, but particularly for these hard substrates and non-reducing ecosystems as they are so understudied. Luckily, I'll be contributing just a bit more to this effort this summer as I continue my research with a generous grant from the Edna Bailey Sussman Fund and with support from Schmidt Ocean Institute. So I'll be conducting analysis of species-level identifications of all the data that I discussed here in order to analyze things like biodiversity and species richness on top of density and high-level composition. And then I'll also get a chance to take a look at those background rocks and compare them to the background rocks I used and the colonization experiment to hopefully figure out if those differences were due to successional stage dynamics or something like seasonality. I'd like to thank my Capstone Advisory Committee, especially my amazing chair, Lisa Levin, who's been sending drafts back and forth with me from Europe, where she's talking at conferences. Also Greg Rouse, who's in the crowd today. Thank you, Greg. Helping me out with quite a few identifications. Anna Zivian for helping me work in some policy and some management recommendations into my project. I'd also like to thank the Levin Lab, with extra special thanks to Guillermo Mendoza, Olivia Pereira, and Michelle Gurayeb, and also the crews aboard the RV Nautilus and RV Falcor, without whom this research would not have been possible. And then, of course, my amazing NBC cohort members. It's been a great year. And to my family and friends for all their support. And I would love to take any questions.
Amazing.
Thank you.
So I will try to keep my question not too, Asia-y.
Oh boy.
So with your knowledge and your assumptions and what you've learned, how do you think that plastic waste and like man-made waste sinking to the bottom of these depths, how do you think colonization happens there? What it looks like, what it's composed of?
Yeah, absolutely. So that was not the main, part of this study, but I can tell you from sorting all of those samples, the number of plastics I found was astounding and also really irritating because it wraps up all the animals. Brittle stars have all these like little tiny legs on their big legs. All the little mites and crabs got stuck in. It was mostly because of the size category that I was doing. Most of the plastics that I saw were filaments, so the type of fibers that come off of like fleeces and stuff. Those end up in the ocean really easily. They just go right through your washing machine. They make it through all of the like cleaning apparatuses at water treatment plants. And so I can't say how they interacted with colonization. I can say that they're definitely there. And I know that there is research on the impact of plastics in the deep sea. It's not super my area of expertise.
Thank you. Your research didn't address interventions, but I think it kind of naturally raises the question of, if we mine a bunch of rocks off the bottom of the ocean, can I dump a different shipload of rocks onto the bottom? And the wood substrates in particular, the fact that it's sort of food at the same time as a substrate, is it possible to think about feeding ocean ecosystems in ways that are meaningful?
Yeah, so that is an interesting idea of sort of like feeding the deep sea. I think generally sort of like bioengineering hacks like that should be approached with caution. But certainly I think there's an interesting perspective to be taken around, like, for instance, I've been thinking about what the impact of all of these wildfires that we're having in California might have on some of the like wood detritus that gets swept into the ocean and what impact that might have on those wood specialist communities. But as far as something like mining that would remove these hard substrates from the seafloor, it's definitely pretty alarming. Just from my research, which is obviously very localized, very small, in a place that probably, hopefully, will never be mined, there were those distinct assemblages across substrate types. And so if you were to remove these substrates, especially since we don't know a lot about connectivity, it could have really far-reaching impacts. And just dumping different rocks probably would not be an acceptable replacement. I'm not a geologist, so I see a rock and I'm like, oh, nice rock. But even just the rocks that were looked at for this study had a lot of different characteristics as far as like how bumpy they are, how porous they were, and that had big impacts on what kind of animals could live in all of those nooks and crannies. So yeah, I would say definitely picking up all the rocks on the seafloor is something that should be approached with caution.
So Lisa Levin, your chair,
Yes.
has a question.
Great.
She'd like to know, if it was your own experiment, is there anything you would have done differently in setting it up?
Yeah, absolutely. That's a really great question. So I thought it was a phenomenal experiment that Lisa put on, but definitely if the funding was available and the time was available, I think having multiple timescales so that I could take a look at different successional stages over time would be really exciting. And also, and perhaps most importantly, more replicates would be great. So the statistics that I was able to do were relatively limited because, for instance, the wood samples, I only had 8 replicates, carbonate rock also 8, and then the other hard substrates in particular had much smaller sample sizes. So those mineral-rich rocks, which are perhaps the most interesting since those are the ones being targeted for mining elsewhere in the deep sea, those I only had 2 each. So definitely it would be great to have a few more replicates. That way we can be a bit more certain about what the statistics say about what's significant and what's not.
Amazing presentation. 2-part question. The first one is, how many hours did you spend sorting 25,000 specimens? That's the first one. The second one is, what type of influence do you think that understanding these assemblages might have on industries like deep sea mining as far as how they might approach impacting that environment?
Yeah, absolutely. So for the first one, as far as hours spent goes, a lot. It was pretty standard for me to be pulling 12 to 16-hour days in the lab for the first 3/4 of this quarter. I was really up to the wire on getting my samples sorted. So yeah, it was a long, arduous process, but worth it. And then yeah, as far as how more information on these deep sea communities could hopefully impact industry, ideally, the industry would wait for the science to come out about what communities live where, how interconnected they are, how resilient they are. There's so little that we know about the deep sea, particularly in areas that have contracts out for mining. And so the ideal situation would be to wait for the science, but since that could take a decade, there hasn't been a lot of waiting happening around these processes. The regulations for the International Seabed Authority for mining overseas and in sort of the high seas area, those are set to be finalized around 2023, so very soon. And while the technology for mining isn't quite there yet necessarily to start mining immediately, I haven't seen any signs of people pumping the brakes within industry. Yeah, so they are required to do environmental impact assessments, which they're hoping will be enough to take a look at the environment. But I think most researchers and conservation organizations would definitely like to see more effort put into determining the sort of baselines of the science before people sort of just full steam ahead on mining. So hopefully we will see that, but who knows?
So it seems like complicated work that would be of interest to a pretty niche audience. What kind of ideas do you have for sharing your work with a broader audience and making it more accessible to a broader audience?
Yeah, absolutely. That's a great question. I definitely think one of the biggest roadblocks to sort of conservation of deep sea systems is that not a lot of people know about it, while the research and the technology to conduct that research has accelerated by leaps and bounds in the past couple decades. The public awareness of the deep sea and its ecosystem services and how cool it is has not really kept up with that pace. And so actually, since this is an interdisciplinary program, I actually did conduct a social media science communication campaign alongside my main science research and also some policy analysis work that I did. And the account started at 0 followers, 0 posts, and I thought it did pretty well given the short time period. So generated 10,000 unique impressions, well over 4,000 accounts reached, and 600 accounts engaged for over 1,000 content interactions, and an average weekly growth rate of about 16%, which was actually pretty steady after the initial bump from all my friends and family graciously following the account. And so I found this to be a positive sign that there is interest out there in the deep sea. People do want to learn about it. It is really cool. And so I am hoping that since I was able to get this much traction with a very tight 10-week deadline and while doing active research and other things, that when I'm able to dedicate a bit more time to it this summer through that Sussman Fund and in partnership with Schmidt Ocean and just on my own time, I'm hoping I'll be able to grow it even more. So feel free to give it a follow. I managed to snag the handle Deep Sea Science, which makes me feel very official.